How the body regulates blood pressure is really important because you can see that if you do not monitor blood pressure appropriately, you can either become hypertensive, and that can lead to things like kidney failure, or you can become hypotensive, and that can actually lead to unconsciousness, coma, and death. So there are a number of different mechanisms in place in the body to help monitor the blood pressure and to basically kick off corrective measures if blood pressure starts to go too high or too low. Some of these different types of blood pressure monitors include a number of what are called neuroreflex arcs. And remember again, a neuronal arc is just a connection or a pathway of neurons between some sort of a sensory receptor and the brain and some sort of outgoing effector to make a change. For example, we have sensory receptors called baroreceptors. These monitor changes in blood pressure, and they're located in a number of places like the aortic arch. Okay, so you see some right here in the aortic arch. They're in the carotid sinus, so right up here there's a little bulging in the internal carotid artery called the carotid sinus. We want to have that monitoring point there because this is directly feeding the brain, so we want to know right away if there's a change in blood pressure. And there's also some monitoring baroreceptors that are found in the right atrium of the heart. Now, all of these sensory receptors send information off to the brain, to places like the medulla oblongata, and if we notice any severe alterations in blood pressure, then we're going to have some sort of mechanism put in place, such as some of the ones I talked in the previous lecture about, the different hormones, etc., that we can secrete to help regulate, get the blood pressure back into a normal range. Your proprioceptors, remember those are found throughout your body in places like joints, muscles, ligaments, tendons, etc. They are there to give the brain information about body position. They're not a true monitor of blood pressure, but they're an indirect monitor because if you start having lots of movement, those proprioceptors are going to be firing off more signals to the brain. The brain interprets that as, gee, we're moving more. That's probably going to impact blood pressure, so we better do something to elevate blood pressure to accommodate the extra demand of skeletal muscle tissue, etc. You also have what are called chemoreceptors. Chemoreceptors are located in a number of places. The two principal ones are in the aortic arch and also in the carotid body, in the carotid artery. And they monitor things like changes in oxygen concentrations, changes in carbon dioxide concentrations, pH changes in the blood. These are all indirect indicators of blood flow and thus blood pressure. So they're not a direct measure of blood pressure, but they do assist the brain in coordinating efforts to ensure that we keep blood pressure within a tight homeostatic range around a set point. What about the brain itself? Well, as I said, the medulla oblongata is a huge player in the regulation of blood pressure. You remembered that the medulla oblongata has the cardiac center, so we can speed up or slow down the heart, and we know that heart rate is related to blood pressure. There's also what's called the vasomotor center of the medulla oblongata, and this is a region of the medulla oblongata that is directly responsible for telling arterioles and other blood vessels, hey, it's time to constrict or it's time to dilate, and in that way we can very rapidly impact overall blood pressure and change it as it needs to. Now, you also have higher centers of the brain in the cerebral, not in the cerebral cortex, for example, the hypothalamus and in the limbic system that can also impact blood pressure. Okay, if you remember, limbic system is what is in coordination of your emotions. So if you get into a fear state, that can elevate heart rate, which can also elevate, for example, blood pressure. On a more microscopic level, locally, individual capillary beds can control when they open and when they close down. So you don't have to rely on the brain to tell a capillary bed when it's time to be perfused or filled with blood and when it's time to not be perfused. The principle by which we think that capillaries are able to determine for themselves when it's time to receive blood and when it's not is the principle of autoregulation. The idea is something like this. When a capillary bed is closed down, when it is not perfused, That means the cells out here near this capillary bed are going about their normal metabolic duties. They're consuming oxygen. They're producing metabolic waste products like carbon dioxide and urea and uric acid and creatinine, all these different metabolic wastes. As those metabolic wastes begin to accumulate in the extracellular fluid, these serve as an irritant. They sort of irritate these precapillary sphincters and causes those precapillary sphincters to dilate. And when they dilate, of course, that's going to allow for the flow of blood through this capillary bed instead of having the blood just go through the thoroughfare channel and bypass. So now it starts flowing through the capillary bed here, picks up all these metabolic waste products, delivers fresh oxygen, fresh amino acids, and other nutrients to those cells, and allows for that exchange to occur that we need to happen. Once the metabolic waste products drop off, then those precapillary sphincters are no longer irritated and they will constrict or close back down again. This is called the metabolic theory of autoregulation. So it's really the metabolic activity of these individual cells that impacts when that capillary bed is opened to deliver oxygen. Now, a couple related terms here. When a tissue is deprived of oxygen, when it becomes low in oxygen, we call it hypoxic. Okay? The condition of being without oxygen is hypoxia. That hypoxia can either be short-term, which is not uncommon when you have a capillary bed that's been closed down for a little bit. You can get normal short-term hypoxia. And usually when the precapillary sphincters open up to respond to that short-term hypoxia, they produce a sudden surge, oversurge, if you will, of blood into the area. This is called hyperemia. The best example I can think of this is if you've ever been playing around with a garden hose in your backyard and you pinched the garden hose off somewhere and pressure of the water builds up behind the point where you pinched it, and then you release the pinch. If you notice what happens to the water when it comes out after you unkink the hose, it shoots out with a lot greater pressure and then it levels back off again, right? Well, it's sort of the same thing that happens here in short-term hypoxia. When we perfuse the area, when we open up those precapillary sphincters, initially we're going to get an oversurge of blood into the area for just a millisecond or so. That's that hyperemia. Long-term hypoxia is a much more serious event. This is when, for extended periods of time, we're talking hours or days, a tissue is not getting sufficient amount of oxygen. This can happen, for example, in the heart when somebody has atherosclerosis and they're starting to get a lumen that's very occluded in one of their coronary arteries. So blood flow to part of the heart is severely diminished, and this persists in happening over days, weeks, months. There are some ways in which blood vessels can respond to long-term hypoxia. One way they can is via a process called angiogenesis. And this means literally, genesis means to create or generate. Angio means relating to blood vessels. So we're literally making new blood vessels to try to accommodate the fact that the current blood vessels, they're just not delivering enough oxygen to the tissues. Okay. Last thing I want to talk about in this section is just how the routing of blood can vary or change depending on the type of activity you're engaged in. If you're hanging around on the couch like this couch potato guy here, the demand of skeletal muscle tissue in your body is pretty low. So there's really not a large amount of reason to deliver lots of blood, for example, to your legs. They're just laying there on the couch. They don't really need a whole lot of blood delivery to them. But if you get up off the couch and you go out and play a game of soccer, for example, now your arms, your legs, your heart, your brain have very high demands for blood that are much greater than, for example, the demand for blood in your small intestine. And so as a result, depending on your activity level, blood routing will vary, and the amount of blood going to particular regions of the body will change depending on the type of activity you're doing. So here you can see that blood flow to the legs is much increased when we're active, and that those iliac arteries are much more constricted. They're smaller in diameter during times of rest. So it's really physical exertion that increases perfusion or blood flow to particular regions of the body. And again, Remember, those arterioles are the big players in this because they have a good amount of smooth muscle tissue and because there's a whole heck of a lot of them. So their constriction and dilation can have profound effects on the routing of blood throughout the body.